Thank you for your interest. I'm Xiangcui Wang from the Institute of Pharmacology at National Yang Ming Jiao Tong University in Taiwan. On behalf of our authors, I'm excited to share our recent publication in JCI Insight titled Aldehyde Dehydrogenase 2 Preserves Kidney Function by Countering Alkaline-Induced Metabolic and Mitochondrial Dysfunction. This study represents a collaborative effort between Dr. Zhijing Lin's clinical team in the Department of Nephrology at Taipei Ventura General Hospital and my research team at National Yang Ming Jiao Tong University.
Chronic kidney disease is a progressive condition that affects more than 10% of general population, and Taiwan has the highest incidence and prevalence of end-stage kidney disease. Renal fibrosis is a common pathological feature of CKD. However, currently there are no effective antifibrotic therapies against CKD. Therefore, finding a new therapeutic target for renal fibrosis is crucial. Previous studies show that lipid nephrotoxicity plays an important role in chronic progressive kidney disease. One of underlying mechanisms could be due to the formation of reactive lipid aldehydes, including 4-hydroxynonenal malondialdehyde or alkaline. Among these aldehydes, alkaline is the most reactive form. Alkaline can be produced endogenously through lipid peroxidation. It can be detoxified through glutathione conjugation and excreted into urine. However, when the detoxification pathway is overwhelmed, the free form of alkaline could attack protein to form alkaline-protein conjugates, leading to cellular damage. Besides glutathione conjugation, aldehyde dehydrogenase 2 (LDH2) is another crucial alkaline detoxifying enzyme. Around 30 to 50% of East Asian population has an inactive missense mutation of LDH2 gene, resulting in glutamate 504 lysine mutation. This polymorphism causes LDH2 to be inactive. Individuals with this polymorphism cannot metabolize acetaldehyde after alcohol consumption, leading to an alcohol flushing reaction. Previous studies have shown that this polymorphism is linked to reduced kidney function. However, the underlying mechanism remains unclear. Therefore, our hypothesis is that during the pathogenesis of kidney fibrosis, alkaline can be produced through hypoxia-induced lipid peroxidation. LDH2 deficiency may cause the accumulation of alkaline, leading to kidney injury and then finally kidney fibrosis. Therefore, this study is to explore the connection between LDH2 and alkaline in the development of the kidney fibrosis. The first study we done is clinical study. Through the cooperation with Dr. Lin at Taipei Ventura General Hospital, we recruited 81 CKD patients and collect their kidney samples for LDH2 analysis. At the same time, also collect clinical data for kidney function and the histology of these patients. The results show that significant decrease in LDH2 expression was observed in the fibrotic kidneys. We also found a direct interaction between the kidney LDH2 messenger RNA expression and the eGFR, which is the kidney function indicator, but the inverse correlation between the LDH2 expression and the kidney fibrosis. The IGC staining showed that LDH2 mainly expressed in the renal tubules, and the higher expression of LDH2 was observed in the kidney tissues of individuals with preserved kidney function compared to the CKD patients.
For influence of the kidney LDH2 expression on aldehyde metabolism, we analyzed the urinary alkaline concentration in these patients. Our results show that the individual with lower LDH2 expression has higher urinary alkaline concentration, and the CKD patients with lower kidney LDH2 expression have a poor prognosis. So based on this result, the reduction of LDH2 and alkaline accumulation may contribute to the CKD progression.
In order to confirm our clinical observation, we utilized two different kidney fibrosis animal models. The first one is the surgical model. We performed unilateral ureteral obstruction model. Another one is the toxicant model. Intraperitoneal injection of folic acid could induce kidney injury. We call it folic acid-induced nephropathy, FAN model.
After 7 or 14 days of utilization, we can observe hydronephrosis, tubular injury, and interstitial fibrosis in kidney tissues. A significant increase in alkaline protein conjugation were observed in UO kidney tubule using IHC or Western blot analysis. Interestingly, we also observed a time-dependent reduction of LDH2 after UO. A similar phenomenon was observed in the FAM model.
In order to elucidate the causal relationship between reduced LDH2 expression and the progression of kidney disease, we use LDH2 glutamate thiophospholysine knocking mice in our animal experiment, and the results show that UO could induce much higher interstitial fibrosis and much higher alkaline protein conjugates in the mutant mice compared to the wild-type mice. A similar phenomenon was observed in the FAM model. Those results suggest that altered LDH2 activity and subsequent alkaline accumulation exacerbate kidney disease. In order to understand the underlying mechanism, we employed a cell culture model. Exposing renal tubular cell culture to alkaline to result in the dose-dependent increase in alkaline protein conjugate PKM2 modification on cysteine-358 and the activity reduction. Furthermore, we found that alkaline-induced PKM2 modification could cause the nuclear translocation of PKM2 and also coactivate HIF-1α signaling using immunoprecipitation assay and CHIP experiments. Using a seahorse metabolic analyzer, we found that alkaline could reduce the maximal respiratory capacity and ATP turnover in primary cultures, and we also confirmed this result in vivo. To further explore the overexpression of LDH2 to prevent the kidney injury, we used the AAV gene delivery through subcapsular injection. After injection, we can see strong kidney transduction in both wild-type and the mutant mice. And our results showed AAV-mediated LDH2 overexpression could decrease the alkaline protein conjugates. And also, the overexpression mitigates kidney fibrosis. And furthermore, we found the overexpression restored the PK activity, ATP production, and also suppressed the HIF-1α signaling in UO model.
So let me summarize the result we have. In this study, we found the kidney injury will reduce the LDH2 expression and the LDH2 polymorphism have lower enzyme activity. Both lead to the alkaline accumulation and then will trigger PKM2 modification and nuclear translocation, which co-activate HIF-1 signaling and induce the mitochondrial dysfunction and finally the kidney fibrosis development. So this study highlight the significant role of LDH2 in the kidney fibrosis pathogenesis, and LDH2 could be a therapeutic target for treating CKD. Thank you for watching. If you are interested in the detail, you can read the full content of our publication. If you have any question, please feel free to contact us by email.